Hello and welcome to this talk on our development of an in vivo in vitro dose equivalence library containing data on exposure to inhalable nanomaterial. This work was performed within the scope of NanoRigo project. Human exposure to nanomaterials through inhalation is the most common route in environmental and occupational settings. Safe by Design creates the need of coherent integration of hazard identification, such as exposure to nanomaterial, with tools of risk assessment. Nanoparticle dosimetry data for both worlds have to be conclusively linked. Risk assessment based on in vitro cytotoxicity testing relies on dose equivalence of in vivo and in vitro scenarios. Any in vivo exposure dose and thereof resulting tissue-delivered dose needs a defined procedure of bridging to a corresponding in vitro administered dose and thereof cellular effective dose. Particle-kinetics are the determinants in both scenarios. When you are working with nanoparticles in vitro under submerged conditions and with adherent cells, dosimetry includes that only a specific fraction of your administered particles will interact with your cells, the delivered dose. The transition from administered dose to a delivered dose is determined by diffusion and gravitational settlement with particle size and density as key determinants. These effects are from relevance not only for in vitro conditions, but also for the nanoparticle exposure in vivo via the lung. Inhaled nanoparticles are retained preferentially in the alveolar compartment of the lung. The deposition pattern and intensity vary depending on nanoparticle size and density. To give in vitro toxicity tests and risk assessment clinical relevance, and to allow a correct interpretation of the in vitro results, convincing dose bridging data and reliable procedures are critical. To support this purpose, we developed a dose equivalence library. The database represents a library of cases for dose equivalence of the nanomaterial reaching cells in an in vitro experiment and relating this to the dose reaching human lung tissues during realistic in vivo exposure. The library is pre-filled with a selection of nanomaterial as showcases with different particle sizes, exposure times, and for different alveolar compartments. The equivalence dose was predicted by means of multiple path particle dosimetry model for in vivo and in vitro sedimentation diffusion dosimetry model for the in vitro simulation. The implementation was done in Excel, a table with different showcases giving examples for the dose bridging, demonstrating its high relevance. It additionally provides the potential to serve as a future data repository continuously updated with further data sets. Hence, this dose equivalence library provides guidance for the refinement of risk assessment and life cycle assessment tools, as well as data quality assessment. Its data model provides a minimum data and metadata set for in vivo in vitro dose bridging. The assessed tools and data could be tested against. Such a library can also guide for the in vitro overdose screening to test in vitro experimental data for relevance in real life. And last but not least, dose equivalence metadata such as exposure duration and delivered dose rate support to check for in vivo in vitro coherence of acute, subacute or chronic biological effects of nanomaterial. Thank you very much for your interest in our work. I would also like to express my gratitude to the funding authority Horizon 2020 Research and Innovation Program. Thank you.